Hi, my name is Dana McCormack, and this final project video will be about default and actively induced fates in the developing nervous system. We're quite familiar with cell fates being induced by morphogens in this class. Most commonly, we've seen how certain molecules initiate patterns of body axes. Morphogens are able to activate transcription factors for different cell types. For example, we've seen how sonic hedgehog and BMPs lead to the development of the dorsal ventral neural tube, and how concentration combinations of Wnt, BMPs, FGF, and retinoic acid pattern the early anterior-posterior axis. In many cases, if a morphogen or its receptor is underexpressed or overexpressed, there might be missing or extra features, such as sonic hedgehog from the notochord and floor plate, inducing ventral cell fates and motor neuron innervation. Or the level of morphogen or its receptor can correspond to the over or under representation of cell types, such as knockouts of PAX6 and EMX2 resulting in different allocation of cortical regions. In this presentation, we're going to talk about three situations in development in which developing cells have a particularly interesting default fate. You're likely familiar with this first example. When investigating the fate of ectoderm cells, the animal cap was removed from the mesoderm and epidermal cells developed, leading scientists to believe that something was secreted by the dorsal mesoderm to induce neural fates. However, when animal cap cells were dissociated from each other, neural cells developed. Himani-Berbenloo and Melton also found that neural fates could be maintained in an intact animal cap by injecting activin, a dominant negative approach that blocked receptors on ectoderm tissue. They later found that BMP4 had a similar receptor to activin, and that it was released by the ectoderm cells to its neighbors. Finally, the identities of neural inducers, noggin, corrin, and follistatin, was found by assaying BMP4 inhibitors released by the dorsal mesoderm. Therefore, the default state of ectoderm cells is neural tissue, while epidermal fates are actively induced by BMP4 and repressed to allow for neural tissue again. Another example of default and induced fates occurs with neural crest cells as they migrate. As neural crest cells migrate, they express a receptor for neuregulin-1 called ERBB4 that, when activated, induces a glial fate, specifically Schwann cells. Neuregulin-1 is also called glial growth factor, or GGF. When they arrive at their peripheral location, they differentiate and stop expressing ERBB4. As neural crest cells differentiate into neurons, they begin producing neuregulin-1. Therefore, early arrival crest cells are exposed to a low level of neuregulin and differentiate into neurons, and later arrival crest cells are exposed to a higher level of neuregulin and differentiate to glia. This would suggest that the default state of periphery neural crest cells is neural, and that glial fates are induced. In culture, the removal of neuregulin-1 results in 80% neurons and 20% glia, while the addition of neuregulin-1 results in 10% neurons and 90% glia. This is an interesting result, as the loss of neuregulin-1 doesn't cause a complete knockout of glial fates. It should be noted that there are several factors at play in differentiation, such as morphogens that could act as inducers of other fates besides glia, shown here, or the possibility of some level of differentiation prior to migration. The last example that we will discuss is the fate of all neurons without intervention: death. There is a family of ligand proteins called neurotrophins that bind to a family of receptors called TRK. When bound, these receptors dimerize and phosphorylate, triggering multiple downstream pathways that promote cell survival and growth. Neurons have multiple potential sources of neurotrophins: the cells that they innervate, which is called target-derived or retrogradely sourced, the cells that synapse onto them, which is called afferent-derived or anterograde sourced, from neighboring neurons and glia, which is a peregrine source, or from blood, a circulatory source. More than 50% of developing neurons undergo programmed cell death. Why would we have an evolutionarily conserved process that seems so wasteful? Firstly, apoptosis is a controlled fragmentation of a dying cell, which is much safer than necrosis, in which the soma swells and bursts, which can damage neighboring neurons. Secondly, apoptosis allows for refinement at all stages of development, from refining the ratios of different types of neurons, to erasing body parts that are no longer needed, to removing cells with DNA errors, to making sure targets aren't over-innervated. A developing embryo has plenty of extra neurons so that it can be choosy about who gets to stay. In conclusion, we've discussed how morphogens can induce features in developing tissue, and we've looked into 3 examples in which the default fate of cells might be different than you'd expect. It's important to remember that cell fates are a combination of intrinsic and extrinsic cues that a cell has received during development. Embryonic cells transition from pluripotent to multipotent as they are exposed to directions from the environment, and the story of default states becomes more complicated as they differentiate because of the complex combination of cues that lead to distinct cell types. Thank you.